Part of many plant foods doesn't go to your enzymes, but to your microbiota. Many components of apple fiber aren't broken down by human enzymes. Once they reach the large intestine, some of them become raw material for the microbial community. And in return, the bacteria make molecules that feed the cells of your own gut. And that's not all. What the body did absorb, a couple of hours later, heads off along unexpected routes. Some of the atoms from your lunch are leaving your body right now with your breath. So why do bacteria need your food? And why doesn't lunch have a single final destination? Let's start with what your own enzymes couldn't digest. Imagine you eat an apple. Its sugars are released during digestion, cross the intestinal barrier, and enter the blood. But part of the apple takes a different route. It's a fragment of the cell wall, one of the structures we call fiber. Human enzymes barely change the molecular structure of many kinds of fiber. So it reaches the large intestine. Along the way, the fibers can swell, bind water, and change their physical properties. But many chemical bonds inside these structures stay out of reach of our enzymes. It's not that the fiber is too tough. It's just that breaking a particular chemical bond requires the right enzyme, a tool whose shape matches a specific spot on the chain. And humans don't have the full set of these tools. It seems like the apple fragment will now just keep going and leave the body. But in the large intestine, it's met by a huge community of microorganisms. Different bacteria have genes that let them build enzymes that human cells don't produce. One bacterium latches onto the plant fiber and starts cutting the complex chain into shorter fragments. But on its own, it does only part of the job. These fragments get picked up by other microbes. Some keep taking them apart. Others turn the intermediate products into new substances. So the microbiota doesn't work like a single factory, but like a network of interconnected production lines, where the product of one microorganism becomes raw material for another. This process is called fermentation. Bacteria use fiber first of all for their own metabolism. They get substances and energy, grow, build new cells. Along the way, gases and many other products appear. And among them, 3 small molecules matter especially: acetate, propionate, and butyrate. They're called short-chain fatty acids. They're nothing like the big droplets of dietary fat, which needed bile, micelles, and chylomicrons. These molecules are much smaller and interact well with a watery environment. So some of them are absorbed by the cells of the large intestine. Butyrate is used especially actively by the gut lining cells themselves. For them, it's an important source of energy. Some of the propionate and acetate goes into the portal blood to the liver. The liver actively uses propionate, while some of the acetate can pass into the general bloodstream. So you get an unexpected route. Human enzymes couldn't take apart the plant fiber, but bacteria turn part of its structure into small molecules, which the gut is already able to absorb. That said, this doesn't happen with all fiber. Fiber isn't one substance, but many structures with different properties. Some fibers are actively fermented, others only partially, and some pass through almost untouched. The result also depends on the makeup of the microbiota, and that's different for everyone. So the same fiber is processed differently in different people and yields different amounts of fermentation products. This doesn't mean one person has good bacteria and another has bad ones. The microbiota is a changing ecosystem whose activity depends on the available raw material, interactions between microbes, and conditions in the gut. Meanwhile, our fragment of apple wall has split up. Some of its atoms became part of bacterial cells. Some ended up in gases and other fermentation products. Some atoms became part of acetate, propionate, or butyrate, and the unfermented remains continued on through the large intestine. So bacteria don't give the person all the energy from fiber. They use it for themselves, but some of the products they make become available to the host body too. Where human enzymes end, the chemical work of the microbiota begins. Now lunch's journey is almost complete. Glucose has ended up in the blood. Fat has passed through the lymph. Amino acids have spread out among the tissues. Even part of the fiber, thanks to bacteria, has turned into new molecules. But what will happen to all of this next? A few hours ago, lunch was sitting in front of you. Bread, meat, vegetables, and a bit of oil, all on one plate. Now that dish no longer exists as a single whole. Starch is broken down into simple sugars, proteins into amino acids. Most of the fat is packed into transport particles, and the fiber is moving through the gut, or has already gone to the microbiota. After absorption, the molecules arrive at a metabolic crossroads. They can be used right now, built into new structures, stored for a while, or gradually removed. And these aren't four separate final destinations. The very same molecule can, over time, go down several routes. Let's start with glucose. Through the portal vein, it goes to the liver, an important distribution hub, which can hold back part of it, change its route, or let it pass on into the general bloodstream. But glucose doesn't turn into pure energy. Inside cells, it goes through a chain of chemical reactions, and part of the energy locked in it is transferred onto ATP molecules, a universal carrier of readily available energy for many cellular processes. The atoms of the glucose itself don't simply disappear. With full oxidation, its carbon ends up as part of carbon dioxide. The blood carries it to the lungs, and some of the atoms of your recent lunch leave the body with your breath. And glucose doesn't have to be spent right away. It can be set aside for later in the form of glycogen. Liver glycogen helps keep the blood sugar level steady between meals, while muscle glycogen is used mainly by the muscles themselves. Dietary fat has its own route. Most long-chain fats leave the gut inside chylomicrons, pass through the lymph and enter the blood, where a special enzyme helps release the fatty acids. Some tissues use them as fuel. Others build them into cell membranes and signaling molecules. The excess can be stored for a while in fat cells. But even that isn't the final stop. When needed, the store is taken apart again, and the fatty acids return to the bloodstream. Amino acids don't head to one place either. Some are used first by the gut and liver. The rest are distributed among the tissues and go toward enzymes, receptors, antibodies, and renewal, including of muscle proteins. The body has no separate storehouse for spare amino acids. If their carbon part is used in metabolism, the nitrogen has to be removed. In the liver, it's turned into urea, which enters the blood, is filtered by the kidneys and leaves with the urine. Not everything that was on the plate manages to go through these routes within a few hours. What's undigested keeps moving through the gut. Part of the fiber is processed by the microbiota. Water and some of the fermentation products are absorbed, and the rest leaves the body later. That's why lunch doesn't have a single final destination, and a few hours isn't a strict physiological deadline. The speed depends on the makeup and size of the portion, the work of the stomach and gut, and many individual factors. What to take from the finale? Fiber isn't there just to check a box. By fermenting it, bacteria produce butyrate, which directly feeds the cells of your gut. So varied plant food feeds not only you, but also your microbiota, and through it, the gut wall itself. And the second thing, what you eat doesn't have a single fate. A molecule's energy can be handed off to other processes, and its atoms can be stored for a while, built into new structures, or gradually removed. So the point isn't in one magic food, but in the overall balance of what comes in regularly. It all began even before the first bite. The brain noticed the food and started the preparations. The teeth ground it up. The throat switched the paths of air and food. The stomach mixed the contents and protected its own walls. The pancreas, bile, and intestinal enzymes took apart the nutrients. The intestinal barrier let the suitable molecules through, while the microbiota handled what human enzymes couldn't break down. The food's journey through the digestive system is almost complete. But the story of its molecules goes on. Food becomes part of you not because it stays inside whole. The body takes it apart, reassembles it, and distributes its atoms between the life of cells and the outside world. This was the season What Happens to Food Inside You. Subscribe to BioCode Lab, where the complex becomes simple.